Hello and welcome. I am Volker Schweikart, Product Manager for Coherent Raman Scattering Microscopy here at Leica Microsystems, and today I'm going to show you what this new way of doing label-free chemical imaging can do for your research. And all of that in just 5 minutes. So let's get started. So to put this new label-free chemical contrast into perspective, let's take a look at some of the popular contrast mechanisms in optical microscopy. In fluorescence microscopy, contrast is typically achieved by labeling one or a few molecular targets with fluorophores. And so in this way, you can get a highly specific, but also a somewhat limited view of your sample. Now, fluorescence has more to offer than just intensity-based information. The lifetime of the fluorescence process provides additional functional information on your sample, and this is exploited in various biosensors. Then we have additional nonlinear optical contrast mechanisms available, such as second-harmonic generation. These typically provide contrast based on some of the biophysical properties of the sample, and so they can, for example, used to visualize biopolymers such as collagen. And then we have chemical contrast that arises from the characteristic vibrations of the molecular bonds that are present within a specimen. And so this provides information on the major biochemical constituents of the sample, where these are localized, how they move, and how they are metabolized. And at Leica Microsystems, we firmly believe that the correlation of all these different contrast mechanisms provides highly synergistic information that is greater than the sum of its parts. Now, how do we achieve this label-free chemically specific image contrast? And the answer is coherent Raman scattering. In coherent Raman scattering, we shine two laser beams onto our sample, a pump beam and a so-called Stokes beam. And when the frequency difference between these two laser fields exactly matches the vibrational frequency of one of the molecular bonds, then these molecules are excited to vibrate very efficiently. So when we couple these two laser beams into a laser scanning microscope and focus the light down onto our sample, then only the molecules in the laser focus are excited. And as a result of this excitation, the light that is being scattered by the molecules is altered, and we can detect these changes. And in this way, as a result, we get a very highly chemically specific and high spatial resolution microscopy technique that is termed coherent Raman scattering. And the instrument with which we do this imaging is the Leica STELLARIS 8 CRS. So what can our customers do with label-free chemical imaging on the STELLARIS 8 CRS? They can do live imaging in cells and organoids without any need for labeling, with zero photobleaching and with minimal phototoxicity, and yet with a very high degree of chemical specificity. The movie shown here is a time-lapse showing contrast from both proteins and lipids in a single shot. So this provides a nice overview of the major biochemical components in this sample. We can immediately discern individual cells. We can see the cell nuclei as dark areas in this contrast. We can appreciate the overall organization of the organoid sample. And at the subcellular level, we can follow the dynamics of individual lipid droplets. So this type of imaging can be done fast at acquisition speeds up to video rate. Our customers can explore complex samples, such as this bone tissue, using a multimodal imaging approach that combines biochemical, biophysical, and fluorescence contrasts. So this is done by combining multicolor coherent Raman scattering imaging with additional nonlinear optical signals like second-harmonic generation, and adding in visible fluorescence using the confocal capabilities of the STELLARIS microscope platform. We can do this type of imaging in a wide range of samples ranging from cells to tissues and even inside small intact model organisms, such as inside the eye of this intact zebrafish, where we can visualize the individual layers and cells of the retina at cellular and subcellular resolution. So with this type of imaging, we have several research areas that we can support. With coherent Raman scattering, we have an entirely new window for probing disease mechanisms and monitoring responses to drug treatment. In cancer research, for example, we can characterize the cellular heterogeneity that is present in tumors, and we can monitor how these cell types respond metabolically to drug treatment. We can work in such cellular model systems like this cancer spheroid shown here, or we can do this imaging in tissue specimens. In pharmacology, we can ask, where does my drug go? So shown here is in yellow a small molecule drug that has been compartmentalized inside vacuoles in the cells that it has interacted with. And the cellular background is shown here in the protein and lipid contrast shown in magenta and cyan. We can support medical research at the basic, preclinical, and translational level. So in this example of a brain tissue from a mouse model of Alzheimer's disease, we can visualize both healthy and pathological structures based on their different vibrational frequencies that reflect the different biochemical and biophysical properties. And so in summary, we really have a great multimodal discovery instrument that can support a wide range of research applications. But we can even go beyond these insightful images. Coherent Raman scattering can be used to perform vibrational spectroscopic imaging. So by tuning the frequency difference between the pump and the Stokes beams, a wide range of vibrational frequencies can be addressed sequentially. So we take an image at each vibrational frequency, and in this way we build up a so-called hyperspectral image cube. So in this way, an entire vibrational spectrum becomes available at each image pixel, and this can serve as a basis for a very detailed biochemical analysis. These spectra can also allow for powerful spectral unmixing algorithms that can provide a quantitative spatial concentration maps of the major molecular species that are present in the sample. So with this, I hope I've sparked your curiosity. I hope you will go explore some of the fantastic content that we have provided for you on our product webpage, where lots of application examples are highlighted, and you can download a brochure and an application note with even more info. We have also a dedicated space for Coherent Raman scattering at Leica Science Lab with application stories and up-to-date publication list that gives you a better feel for what's currently going on in the field. So thank you for watching, and I'm very happy to hear your feedback and questions.